Everybody, my name's David Eisner and I'm the Deputy President of the Physiological Society. I must say I'm slightly bemused to wonder exactly why I'm standing here at a meeting of women physiologists. Before I came here I could think of two explanations, one very positive and the other rather negative. The positive one is that our diversity champions, Sue Dukas and Rachel Tribe, quite properly feel that we should gender balance people who talk at meetings, and therefore I'm here. The more negative one, the more malign one, is that our organizer here wants me to undergo and sort of appreciate what it's like to be part of a token minority. But fortunately, listening to Tilly's talk today, I realized the real reason I'm here. She talked about the unsung heroes, the unsung founders of the Physiological Society, the wives of the physiologists. I realized if I have any chance of reaching posterity in 100 years' time, the only way I'll do it is as the husband of this lady here. So perhaps that's why I'm here. All that nonsense being said, it's a real pleasure for me to introduce Kim Barrett. As she's already said, Kim grew up in London. She did her BSc and PhD degrees at University College London, and her PhD was under the supervision of Fred Pearce. She studied and discovered the heterogeneity of mast cells and the importance of this heterogeneity. In 1982, she moved to the NIH and carried on working on mast cells, but now very much in the context of the gut. In 1985, she joined the faculty at the University of California, San Diego, where she began her work on intestinal transport. She rose through the ranks very quickly and became a full professor in 1996. As I'm sure we'll hear, her scientific work has focused not only on the mechanisms underlying intestinal transport, but importantly how inflammation affects them. And she studied both the good and bad effects of bacteria in the gut. She's not, however, only a scientist. Those of you who've been to physiology meetings will have heard her singing with her group GI Distress. In fact, to use the vernacular, that sounds like a very crap name for a band. I first met Kim when I applied for a position and she was on the interviewing team. I didn't get the post, but there you are. So, Kim has had countless accolades and awards. She's given the American Physiological Society's Davenport Lecture. She's also been elected a foreign member of the Swedish Royal Society of Sciences. She served the American Physiological Society both as chair of its publications committee and as president. Now, we're celebrating quite rightly the fact that it's 100 years since women were elected to our society. The Journal of Physiology, our premier journal, is almost 140 years old. Now, if I'd been giving this introduction a year ago, or even a couple of months ago, I'd had to bemoan the fact we've never had a woman editor-in-chief of this august journal. But I'm very pleased to say, as many of you will know, that Kim is not only our first female editor-in-chief, but she's also the first ever editor-in-chief from outside the United Kingdom and Ireland. And I think that testifies for her eminence. I could talk longer, but you'd much rather listen to Kim. So it's my great pleasure now to invite Kim Barrett to give the 2015 Bayliss Starling Prize Lecture, Endogenous and Exogenous Regulation of Transport Function in the Intestinal Epithelium, building on the legacy of Bayliss and Starling. Kim.
So I really want to thank David very much for that warm and witty and kind introduction, and it's a huge honor for me to be here and have the opportunity to give this lecture. In fact, when I got the letter inviting me to give the lecture, I didn't quite believe it. I thought it was sort of a hoax. It's especially an honor for me to give this particular lecture that is named for Bayliss and Starling because of the contribution that they made to our understanding of physiology. As I'm sure everybody in the audience will know, among many things that they did, and many of those things were done at UCL, my alma mater, so that makes it even more significant. They were the discoverers of secretin, which was really the origin of understanding about hormones. And in fact, Starling was the one who sort of coined that term. What I didn't really know, even though I teach GI physiology, and every year when I would give the lecture on GI physiology, I would talk about this discovery of secretin as the first hormone. I didn't know that this work was published in the Journal of Physiology, which is a terrible omission for me to admit. But I do actually direct you to a wonderful one-page classical perspective that was written by Barry Hurst at the University of Newcastle that basically puts in context that original publication. And it's a particularly wonderful piece. And actually, I saw Barry last week because I was invited to Newcastle to give a talk, and I thanked him for this great article. It gives a sort of verbatim discussion of the actual discovery by Martin, who was present and later wrote the obituary for Starling that was published in the BMJ. And I won't read the whole thing because it'll take us too long, but it finishes up with, this is very confusing. Oh, you have to have it the right way round, then you have the, yeah, other side. It finishes up with this quote, "It was a great afternoon," perhaps the mastery of scientific understatement. But as Barry says, that the discussion provides fabulous insight into the excitement of the moment. This is one of the slides I've used for years in teaching how the gastrointestinal system is regulated in my first introductory lecture to medical students. And we now take it for granted that there is endocrine regulation of gastrointestinal function as one of the four main mechanisms, not only, of course, regulation of the GI tract, but virtually everywhere else. And it's quite amazing that it's really only a little over 100 years ago that this whole concept originated, and sort of tells us, in fact, sometimes when you're in the lab you feel like science is moving rather slowly, but in fact it moves really quite rapidly. So with that by way of background, how does my work relate to this theme? I'm going to talk to you today about the intestinal epithelium, or the intestinal epithelium, which I've forgotten how to say it properly, and what it does in the gut. And I thank the previous speaker in the previous session for telling you how terribly important it is and non-boring the enterocytes are. One of the things that the epithelium does is participate in electrolyte transport, and that is to subserve its physiological functions in digesting and absorbing nutrients. And one of the things that wasn't so obvious when I really entered this field, but now is quite clear, is that the epithelium both absorbs and secretes electrolytes to carry out these functions. The thing that really got me into the field in the first place was the translational significance of understanding intestinal epithelial transport. And that is because while my mom would prefer that I not work on diarrheal diseases, that's what I've chosen to do. She says it doesn't make for good conversation at sort of parties at her retirement home, but that's, for good or bad, what I work on. And so understanding transport may allow us to intervene when transport function is abnormal. So, a brief introduction to the intestinal epithelium. As I'm sure most of you know, it's a single layer of cells that divides the inside of us from the outside world. It's a continuous monolayer of cells stretching from the stomach to the anus. And that single layer is really all that's protecting us from the outside. But it's very important that it be a narrow layer, because just as we need a thin interface for gas exchange in the lung, we need a narrow interface to allow us to take up nutrients and deliver them to the rest of the body. The gut is contiguous with the outside world, and it is therefore an important portal not only for nutrients to enter the body, but potentially for things that we'd rather keep out. So in fulfilling its physiological functions, the intestine is continuously absorbing not only the nutrients that we take up in the period after meals, but also a variety of electrolytes, sodium chloride listed here, quantitatively the most important, but also other electrolytes. And being driven by that uptake of electrolytes and nutrients is water. In fact, the gut uses about 8 liters of water per day in providing for digestion and absorption. About 1 liter of that comes from the food and drink that we ingest, but most of it is coming from the body itself. Obviously, I think everyone will recognize that 8 liters does not normally come out at the other end. And in fact, if it does, you'd very rapidly be sick. So most of the fluid and electrolytes that is used in allowing the gut to fulfill its functions is reabsorbed. Nevertheless, there is ongoing secretion by the gut, predominantly driven by the active secretion of chloride ions and water then following, and that allows us to adjust the fluidity of the intestinal luminal contents as needed to subserve the digestion and absorptive process. Other important characteristics of the gut include the fact that there are a variety of different cell types within this epithelium, all originating from the gastrointestinal stem cells that we just heard about. Some of these secrete products that are important in gut homeostasis, including mucus that's secreted by the goblet cells, antimicrobial peptides that are secreted by Paneth cells. As we also heard from in the early stage investigator session, there are endocrine cells within the intestine that secrete hormones predominantly, although not exclusively, into the bloodstream side to regulate function of the system. And of course, secretin, discovered by Bayliss and Starling, is one of those hormones that's secreted in response to changes in luminal conditions. The gut is also a key barrier, as I had implicated, so it has this physiological imperative to allow some things in while excluding, under healthy conditions, toxins, pathogens, and indeed may actually be involved in actively effluxing toxins from the epithelium that the body would much prefer to exclude. And then finally, as we've also touched on this afternoon, the epithelium is in constant communication from birth with a complex community of commensal bacteria that almost certainly are exchanging information with the epithelium and regulating its function. So I'm going to focus particularly today on the transport functions of the gut, but many of those other concepts will also be interwoven in terms of the intestinal epithelial biology. So I wanted to introduce you to 3 of the key electrolyte transport mechanisms that we'll be touching on as we go along. The first of these is electroneutral sodium chloride absorption, which is diagrammed here. So this is predominantly a function of villous epithelial cells in the small intestine, surface epithelial cells in the large intestine, and is quantitatively significant in providing for fluid and electrolyte reabsorption in the period in between meals when nutrients are absent. So it consists of the coupled activity of 2 apically localized transporters in intestinal epithelial cells, DRA, which stands for downregulated in adenoma, which gives you a key as to how it was discovered, but it is indeed an ion exchanger, and sodium hydrogen exchanger, predominantly the isoform NHE3. Working together, these transporters allow for the influx of sodium and chloride in a coupled fashion, which can then be exported across the basolateral epithelium and driven secondarily in a sort of energetic sense by the basolaterally localized sodium-potassium ATPase. So the net effect is that sodium and chloride move across the epithelium and water will follow paracellularly. In the distal large intestine, but not elsewhere in the gut, there is also an electrogenic sodium uptake mechanism that is important in the uptake of the residual fluid that remains as the luminal contents begin to exit the large intestine. So I and others have seen this as an important salvage mechanism whereby the fluidity of the stool is finally adjusted so that excessive amounts of water are not lost from the body. This mechanism is mediated by ENaC, the epithelial sodium channel that is also important in renal physiology for sodium reclamation from the urine. and again driven by the sodium-potassium ATPase. In this case, there's no accompanying anion, and so chloride and other anions, as well as water, will follow paracellularly. The importance of this transport mechanism is illustrated by the fact that in inflammatory bowel diseases such as ulcerative colitis and Crohn's disease, there is specific loss of the ENaC sodium channel expression on the apical membrane, and that is thought to contribute to the diarrheal symptoms that are such predominant features of those diseases. And then finally, on the secretory side of the equation, we have chloride secretion, quantitatively the major transport mechanism that is providing for the addition of fluid to the gut luminal contents. So chloride secretion is more complex molecularly in terms of the number of participants, but it's basically using the same sort of polarized organization of transport molecules to allow for chloride to move from the bloodstream through the cells and out into the intestinal lumen. And in this case, a cation, typically sodium and water, will follow paracellularly. The key constituents of this chloride secretory mechanism include the CFTR chloride channel that is defective in the disease of cystic fibrosis. And of course, its absence will lead to diminished ability to hydrate the gut luminal contents and intestinal obstructions. There are also other chloride channels that serve apparently accessory roles in the chloride secretory process. There's NKCC1, the sodium 2 chloride cotransporter that provides for the chloride uptake into the cell, and this is driven secondarily by the prevailing sodium gradient established by the sodium-potassium ATPase. And there are also basolateral potassium channels which recycle the potassium that's taken up by NKCC1 and allow for maintenance of the driving force for chloride exit. So when these transport mechanisms are put together, we can start to get an idea of what's happening in terms of whole tissue intestinal transport function. So in health, in the small intestine, there is some, although not absolute, segregation of transport properties such that absorptive mechanisms are predominantly localized to the villous epithelial cells, and secretory mechanisms are predominantly localized to the crypt. So as the epithelial cells are generated from the stem cells that are localized to the crypt and the cells migrate out onto the villus, the transport properties of those cells evolve to cause this shift in transport function. And as indicated by the relative size of the arrows, in the villus, the absorptive vector predominates overall, but secretion is also constantly ongoing as needed. And I will touch on that in the first part of the sort of research data that I'm going to share with you. The colon's very similar, other than it doesn't have villi, its morphology is slightly different, and also typically there's not nutrients present to be reabsorbed, although there are high concentrations of short-chain fatty acids which are derived from the bacterial fermentation of unabsorbed carbohydrates. But again, we get this segregation such that the absorptive mechanisms are largely in the surface, and the secretory function is largely segregated to the crypts. Again, the water balance is driven primarily by absorption, but it's likely that secretion contributes to modulating the local composition of the lumen and or may be important in maintaining crypt sterility. So flushing out undesirable bacteria or their products from these relatively stasis-prone crypts. So one of the things that drives that chloride secretion, as we'll see in a moment, are reflexes that involve the enteroendocrine cells that are present within the epithelium and the enteric nervous system. So I'm going to tell 3 stories. And I'm glad that Susan referred to storytelling. This is probably the only point at which my presentation will conform to all those wonderful rules she gave us about talking to people. But I am going to tell 3 stories. And they'll be somewhat abbreviated stories. I will apologize in advance if I don't give all of the underlying data. We might be here all night, and I'm sure with me being the only person standing between you and the drinks reception, you wouldn't want me to go on all night. So the first story I'm going to tell you is, quote, the wisdom of the epithelium, to sort of paraphrase another famous physiologist, Walter Cannon, who talked about the wisdom of the body, to explain to you why I think that diarrhea is not a permanent state. And I'll explain that as we go along. Secondly, I'm going to touch on those commensal bacteria and other components of the microbiota and how they regulate and protect epithelial function. And then finally, talking about the bad bacteria, how a pathogen, in this case Salmonella, subverts epithelial function to cause diarrhea. So story number 1, which relates basically to the intrinsic regulation of epithelial chloride secretion. And I will make the disclaimer, but this is the most compressed story with the least actual data presented. So another thing that you need to know about chloride secretion in the gut is that there's basically 2 major signaling pathways that lead to the activation of chloride secretion. The first is mediated by cyclic nucleotides, and it just so happens that cyclic nucleotides, and particularly cyclic AMP, lead to large, sustained chloride secretory responses that continue as long as the agonist is present. So neurotransmitters such as VIP or prostaglandins that are produced in the setting of inflammation, they turn on secretion and it stays on. On the other hand, there are also calcium-dependent signaling responses that are small and transient. And for that reason, initially were somewhat ignored. But it's now felt that those calcium-dependent responses are actually very important for 2 reasons. First of all, they fine-tune the response to cyclic nucleotides because there is in fact a synergism between calcium and cyclic AMP in stimulating chloride secretion. And secondly, they are evoked by physiological stimuli such as distortion of the epithelium, intestinal distension. Every time a food bolus passes along, it's felt that there's a little burst of secretion to lubricate the gut and allow that to pass without gastrointestinal injury. So, what are some of the characteristics of a response that would serve that temporary lubricating function and the fine-tuning response? You want them to be small, you want them to turn off, you don't want to turn on chloride secretion every time a particle of food abrades the epithelium and then not turn off, because if they didn't turn off, you would constantly be having diarrhea. So why do we think that happens? Well, the prototype of an agonist that causes calcium-dependent chloride secretion is acetylcholine, released from enteric nerve endings, binds to an M3 muscarinic receptor on the basolateral membrane of crypt epithelial cells in the small and large intestines, and evokes an increase in intracellular calcium. And not shown here, this in and of itself is capable of activating chloride secretion. However, even if calcium increases are sustained artificially, there is also inhibitory signaling that cuts in immediately thereafter. And in a series of papers, we showed that this inhibitory signaling actually involves a complex set of signals that crosses the membrane several times. There's recruitment of a soluble tyrosine kinase called PIK2 that is activated by calcium, activation of membrane-bound matrix metalloproteinases that can cleave membrane-bound forms of growth factors such as transforming growth factor alpha, and then a transactivation of a receptor tyrosine kinase, EGFR, that then in turn culminate in inhibitory signaling via the apical membrane. And we think that this signaling is directed particularly at those accessory calcium-dependent chloride channels. In the course of this work, however, we also discovered that EGF itself, the natural agonist for the EGF receptor, is also an inhibitor of intestinal epithelial chloride secretion, and particularly calcium-dependent chloride secretion. And so this may also be an intrinsic braking mechanism that limits the ability of the epithelium to activate chloride secretion, because EGF receptor ligands are naturally present within the gut as one of the primary mechanisms that causes epithelial proliferation and differentiation. However, unlike the mechanism that was promoted by carbachol or acetylcholine, when EGF binds to its receptor, it recruits a different signaling mechanism that is directed towards the basolateral membrane, and via recruitment of phosphatidylinositol 3 kinase, production of specific phospholipids, and recruitment of a particular isoform of protein kinase C, there is a direct inhibitory effect on potassium channels. So why is there this distinction between how does the cell know that the EGF receptor has been activated by its bona fide ligand EGF or transactivated by carbachol? We know that the downstream signals that are recruited are distinct, as indicated by the blue and the pink arrows, and while the end result is inhibition of chloride secretion, the mechanisms are also distinct. And at least in part, this appears to relate to the specific characteristics of the EGF receptor, which we now know signals through differential patterns of the key signaling tyrosine residues within the intracellular carboxy terminus of the protein. And I won't go through these different tyrosines in detail, but suffice to say that different ligands or transactivation of the receptor may recruit different signals. Another part of the story comes from the knowledge that the EGF receptor is actually a member of a family of receptor tyrosine kinases called the ERB receptors. And depending on the specific census of ERB receptors that are present within different cells, you can get both homo and heterodimerization of the receptors and recruitment of a different set of downstream signaling effectors by virtue of those receptor recruitments. Also interesting here is ERB2, which, despite being the most catalytically active of the ERB receptors, is an orphan receptor with no known ligand. So by studying these signals, we've now come up with the following scenario for why there are distinctions between the downstream signals recruited by EGF and carbachol. So when carbachol acts on its receptors, it does lead to the release of TGF-alpha, which can activate the EGF receptor in much the same way that EGF itself does. But carbachol also activates receptor tyrosine phosphatases, including protein tyrosine phosphatase 1b, that specifically removes the phosphates from two of the key signaling residues. If we knock out PTP1B, then that results in rephosphorylation of those receptor residues and the recruitment of signaling typical of EGF. So sort of summing up this story and its implications, calcium-dependent agonists stimulate but then restrict intestinal chloride secretion via the EGF receptors. EGF receptor ligands also limit calcium-dependent chloride secretion but don't serve as agonists of the process. And in addition to telling us perhaps why we're not all living in a permanent state of diarrhea, which would not be very convenient for meetings such as this, especially with the limited bathrooms here in Hodgkin-Huxley House. I would like to think that these overlapping but distinct mechanisms of inhibition may allow chloride secretion to be restrained without risking excessive mitogenic signaling that would then in turn predispose to intestinal cancers. Okay, story number 2. And this focuses on commensals, but specifically a particular class of commensal bacteria that are present in the gut, the so-called probiotics. So, probiotics. They are commensal microorganisms that have been selected for their apparent benefit for health for a variety of conditions. And initially, those really stretching back for millennia, where these have been added to foods, we had only anecdotal evidence that they were good for you. They then started to be good for you for just about anything. So you only have to watch the television for a little while to see these things advertised, yogurts that contain probiotics and so forth, and they're good for everything. Gastrointestinal infections, IBD, digestive disorders, autism, depression, cardiovascular disease, you name it, the microbiome and therefore probiotics has something to do with it. Patients love them. They've been used for millennia. They're natural, so people like them, particularly in Europe. And I would say that we are having emerging insights into their mechanisms of action. So some of the work that we've contributed in this sphere has really asked the question of probiotics or commensals are in a position to interact directly with the epithelium. Might they interfere or regulate epithelial function in a beneficial way? So the first thing we looked at was looking at chloride secretion in cell lines, and we treated these cell lines with a probiotic preparation that consisted of 2 different probiotics in pink. For today's purposes, doesn't matter what they are. And then we infected the epithelium with a pathogen, in this case enteroinvasive E. coli, that we know stimulates chloride secretion, at least in vitro. And if you pre-treated with the probiotics, this stimulatory effect on chloride secretion was significantly abrogated. Now, in part, that appears to be due to the fact that the probiotics actually interfere with the ability of the pathogen to invade into the epithelial cells. It's a physical barrier that's preventing infection. There's also another beneficial effect, which is we know that pathogenic bacteria often reduce the barrier function of the epithelium, as shown here by measuring resistance. And when you pre-treat with probiotics, that other deleterious effect of the pathogen is at least in part reversed. And what you'll also see here is that probiotics alone in control cells actually cause a significant improvement in the barrier function of the epithelial cells. But this isn't just about interfering with other bacteria and interfering with them getting into the cells. It's been known for many years that a variety of inflammatory cytokines, in this case gamma interferon, can also cause a decrease in intestinal barrier function. And when we treated with interferon, we saw a decrease in barrier function. When we pre-treated with probiotics, this barrier decrease was almost completely abrogated. So the epithelial cells are being triggered by the probiotics to be protected against an injurious or inflammatory signal. And similarly, interferon is known to significantly suppress the ability of the epithelium to secrete chloride ions. So here, the interferon-treated condition. If we pre-treat with probiotics again, this damaging effect is almost completely lost. So a direct effect of the bacteria. You have to have live probiotics, and that's why probiotics in yogurt and similar preparations where they are still alive is thought to be so beneficial. If we use just DNA from the probiotics or heat-killed probiotics or just the products that they secrete, none of those things were protective. So probiotics can have beneficial effects on the epithelium directly. So of course, all this work in vitro is very nice, but in fact you would like to know what happens in living animals as well. And these studies were conducted in a mouse model of colitis that is presumed to occur secondary to a defect in epithelial barrier function. So if you take mice that lack a protein called MDR1A, they get a spontaneous colitis, and it's been shown to be dependent on the epithelial defect rather than a defect in immune cells, and it depends on intestinal flora. So when we looked at these mice, whether it was the weight gain that was experienced by these animals during the period that they were getting colitis as a measure of their overall health, mice treated with placebo, the knockout mice had a significantly diminished weight gain because they were getting sick, and treatment of these mice with probiotics preserved their rate of weight gain. If we looked at the barrier function of the gut by looking at bacterial translocation across the gut, that bacterial translocation was significantly reduced in animals that received probiotics. And you could do this either in a pre-treatment before they started getting sick, or even in mice where colitis had already begun. And it also partially ameliorated transport dysfunctions that you can measure in the excised gut from these animals in terms of their ability to secrete chloride in response to forskolin. And then looking at the sort of gold standard, well, are they actually less diseased? We measured the incidence of diarrhea in these animals, and this was done in animals where colitis had already begun, where 100% of them were exhibiting diarrheal symptoms, and over a 2 or a 4 week period, we saw a gradual diminution in the incidence of diarrhea. So we think that probiotics can mimic the beneficial effects of commensals at the epithelial interface by directly interfering with the pathogenic bacteria, by blocking adhesion or invasion of those bacteria, by improving barrier function, reversing transport dysfunction, antagonizing the effects of inflammatory mediators. So to summarize this story, probiotics can ameliorate barrier and transport functions induced by pathogens and inflammatory cytokines. They reduce diarrhea and dysfunction in a colitis model. And indeed, these effects of probiotic microorganisms may contribute to their efficacy in a variety of digestive diseases and perhaps other diseases where epithelial barrier dysfunction have been implicated. So, story 3, very quickly. Salmonella, which is an important foodborne pathogen, but surprisingly, its ability to cause diarrheal symptoms was really not very well understood. So foodborne non-typhoidal salmonella are among some of the most prevalent causes of infectious diarrhea. Even in developed countries like the UK or the US, the disease may be severe enough to cause death, particularly in the young, the elderly, people who are institutionalized. There's rapid emergence of antibiotic-resistant strains of these bacteria. And as I said, the pathogenesis of salmonella diarrhea, and in fact that caused by invasive pathogens in general, had been relatively poorly understood. So I've been working with two wonderful colleagues in infectious diseases in San Diego to try and redress this balance. And the advantage that they brought to the table was a mouse model of salmonella diarrhea. Prior to this, if you infect people with salmonella, they get diarrhea. If you infect calves with salmonella, they get diarrhea, but mice did not. And so we didn't have a good small animal model, and that really impedes an understanding. The part of the reason for mice not getting diarrhea appears to relate to the fact that most mouse strains that we use in the lab are defective in a protein called NRAMP1. And so Josh Fira and Don Guiney, my colleagues, made a mouse that was cogenic for wild-type NRAMP1. And if you infect those animals with salmonella, they do get diarrhea, whether you measure that just using an analog score or you measure the percentage of water. And the diarrhea requires that the bacteria are competent for epithelial invasion. If we use a mutant, you don't get the diarrheal symptoms. So the question we then had is, why do they get diarrhea? So we're a transport lab. We measured transport in tissues from these animals. And much to our surprise, rather than seeing an increase in chloride secretion, such as you would expect in cholera, we actually see a diminution, both in transport under basal conditions in tissues from mice infected with wild-type bacteria, or in response to the cyclic AMP-dependent agonist forskolin. And that's true in both the proximal colon and the distal colon. So this doesn't explain the diarrhea at all, but nevertheless, we wanted to understand why it happens. And in fact, if you look at CFTR, that important chloride channel that drives the majority of chloride secretion by confocal microscopy, normally in control tissues, it is localized to the apical membrane of crypt epithelial cells, as shown here with double staining for CFTR in green and vilin, an apical epithelial marker in purple. You get this white co-localization of both proteins. In tissues from animals infected with wild-type salmonella bacteria, the vilin is still there, so the apical membrane is intact, but CFTR appears to be lost from the apical membrane. I don't have time to go into the controls, but suffice to say that this loss from the apical membrane is not because CFTR is not being produced. It appears to be mislocalized or perhaps internalized in infected mice without a change in expression. But it still doesn't explain why they get diarrhea. So then we thought, well, maybe it's nothing to do with an upregulation of chloride secretion. Maybe it has to do with changes in the absorptive machinery. So we looked at ENaC, which is, as I told you, is an important component of electrolyte absorption in the distal colon. And indeed, ENaC is selectively lost, focusing on these two panels here, from the surface epithelial cells in wild-type infected animals. And in this case, there is a change in expression, both by Western blotting and by at the RNA level for one of the ENaC subunits. Similarly, DRA, the chloride bicarbonate exchanger that is normally involved in the proximal colon in absorption of sodium and chloride across the apical membrane of surface cells. Again, it's an apical protein, co-localizes with vilin, in this case in the surface, but not the crypt cells. In wild-type infected tissue, DRA expression is essentially not evident while vilin remains intact. And this is also reflected at the level of expression by either Western blotting or mRNA measurements. We wanted to understand how this happens, and it's been known, of course, for very many years that salmonella results in an acute inflammatory response with infiltration of neutrophils into the tissue. It's also been thought for many years that those neutrophils are actually required for origination of the diarrheal symptoms. But if we took mice in which neutrophil infiltration was actually blocked by knocking out the receptor for interleukin 8, both the downregulation, the expression of DRA that we see, or the suppressed expression in infected tissues, was still present, whether or not IL-8 receptor was there. And there's also a significant suppression of ENaC expression that was independent of the IL-8 receptor. So what's happening in these animals? Well, we now think that infection with salmonella is having an impact on the ability of the epithelial cells to differentiate and produce the mature surface colonocytes that are required for the absorptive process. And one of the reasons that we're using this as our working hypothesis is if you look at proliferation in the colon from these animals, under control conditions or just in mice that are pre-treated with an antibiotic, kanamycin, we see, as expected, that proliferative cells are really restricted to the base of the crypts, and the depth of the crypts is not altered. When we infect with wild-type Salmonella, there's an increased number of proliferating cells. They're seen at much higher levels of the crypts, and the actual depth of the crypt is increased. So there's an increased turnover of the epithelium, which we think may contribute to disease. And in fact, the IL-8 receptor was also not required for these impacts on epithelial dynamics, whether we looked at crypt length or the number of proliferating cells that were stained for Ki67. So our working hypothesis is that Salmonella infection impacts on the program of differentiation within the gut, where the stem cells within the crypt can choose to become either the absorptive enterocytes that are important in expressing DRA and ENaC, among other markers of differentiation. There's also a different lineage choice that generates goblet cells, endocrine cells, and other secretory lineages. And at least our preliminary data suggest that Salmonella infection is increasing passage through this pathway at the expense of the absorptive pathway. So to sum this up, in Salmonella infection, there isn't a big increase in chloride secretion. In fact, chloride secretion appears to be diminished. But at the same time, the capacity for absorption likely results in fluid accumulation in and of itself, because the colon is normally the important site where the stool is dehydrated. If that doesn't take place, then diarrheal symptoms can ensue. We also do think, however, that the reduction in secretory capacity in the crypts may have implications for host defense, because as I mentioned earlier, the chloride secretion and the accompanying fluid that normally enters the crypts may be very important to maintain crypt sterility and protect that important stem cell niche. And one of the reasons why I suggest this is this rather simplistic experiment that we did many years ago and never actually published, but I think it's instructive, and we really should return to this project. So in this experiment, actually what we did was we treated epithelial cells with cholera toxin, which is one of the strongest stimuli of chloride secretion that we know, and it's also an irreversible stimulus. And if you then infect these epithelial cells, which are polarized from the apical surface with Salmonella bacteria, wait for an hour, then kill the extracellular bacteria and lyse the cells. If you then do serial dilutions, you can see that cholera toxin significantly impeded the ability of the bacteria to invade. So very circumstantial evidence, but suggestive that chloride secretion can actually be protective in the setting of invasive bacteria. So to sum up, diarrheal symptoms in Salmonella-infected mice are accompanied by changes in iron transport. Expression, both some but not all of absorptive transporters, is downregulated, requiring bacterial invasion. This is accompanied by a hyperplasia of the colonic crypts, and ultimately understanding how Salmonella impacts transport function may improve our ability to treat the associated diarrhea. And this is a real-world problem. This is simply one outbreak of Salmonella disease in the US that occurred in 2009. It says in this news piece that 9 were killed. Actually, eventually, 11 people died in this outbreak, and over 1,000 people were sickened. And it all came down to peanut butter. And unfortunately, this company, the Peanut Corporation of America, supplied peanut butter all over the country to hospitals, to daycare centers, to schools. So it wasn't just an isolated area, it was all over the country. And the chief executive of this country was recently in prison for life because they had evidence that he knew that the peanut butter was tainted, and he said, "Ship it anyway." That was his quote in the email. This is kind of what keeps me going to work, though, not worrying about peanut executives, but people worldwide who suffer from diarrheal diseases. This is a classic picture of an individual in Thailand who recovered in a cholera epidemic, and the two nurses who kept him alive, and the IV fluid bottles that were required to resuscitate him during his illness. So diarrheal diseases are real-world things. They cause many, many, many deaths, not only in the third world, but especially in places where we can't assure clean food and water supplies. So I hope I've convinced you that intestinal homeostasis requires precise control of transport and barrier function, that both endogenous and exogenous factors, not quite the hormones of Bayliss and Starling, but certainly a variety of different factors, are continuously modulating the physiological properties of the epithelium, that some effects of bacteria, as one example of an exogenous factor, can be beneficial and may provide insights into the mechanism of action of probiotics. And ultimately, if we understand this area better, we may be able to intervene in diseases that are a major worldwide scourge. So it just remains for me to thank you for listening and for staying this late to hear my presentation. The many members of my lab, both past and present, who've contributed to these studies, our collaborators, of course, the various NIH government and private agencies that have supported the work over the years. And I happen to know, I think that maybe a couple of my friends in the US are watching this, so hello to you, because it was wonderfully webcast. And thank you very much to the Physiological Society for this amazing honor.
Thank you very much, Kim. And I'm such a dedicated fan of peanut butter, but nevertheless. I'd like to thank you on behalf of everybody for a wonderful talk. I'm particularly impressed by the fact that it's a complex system you've been talking about. Anyone such as myself will balk to attempt to talk about intestinal function to the uninitiated such as myself, and I think you've done this beautifully. You've done it by demonstrating the importance of integrated physiology, also cellular signaling and cellular physiology. And you put it all together while suppressing any tendency towards verbal diarrhea. So, very much for that. So, as a token of our appreciation, I would like to present this to you. But I should say, for those of you who want to revise what Kim talked about today, the video will be released. And I believe you're writing this up. This is a little prompt.
Yes.
For the Journal of Physiology. So we look forward to reading that, and we present you with this token of our appreciation and our congratulations for the Bayliss Starling lecture.
Thank you.
Thank you
Really
very much.
beautiful. Thank you so much, Richard. Thank you. I think my husband wants to get a picture.